Carbohydrate metabolism, glycogenolysis. In this slide presentation, we will discuss the process and regulation of glycogenolysis, which involves the cleavage of glucose units from an existing glycogen chain. This process is catalyzed by the enzyme phosphorylase, which cleaves the alpha-1,4 bonds of the glycogen chain. Debranching enzyme cleaves the alpha-1,6 bonds of the glycogen branches. Like with glycogenesis, it is important to discuss the metabolic environment promoting the process of glycogenolysis. In contrast to glycogenesis, glycogenolysis is a process of tissue catabolism versus a storage process. This catabolic process of glycogenolysis is used to promote ATP synthesis and mobilization of glucose in a fasted state. During glycogenolysis, glucose is cleaved from the glycogen chain as glucose 1-phosphate, as seen here. Glucose 1-phosphate is then converted to glucose 6-phosphate, which can either undergo glycolysis or be converted to glucose. Glucose 6-phosphatase is needed to convert glucose 6-phosphate to glucose. Glucose 6-phosphatase exists in the liver, but not in the muscle. Therefore, the liver can aid in regulating blood glucose because it can convert this glucose 6-phosphate to glucose, and then that glucose can enter the bloodstream. This diagram illustrates the steps of glycogenolysis with first the cleavage of glucose as glucose 1-phosphate, and then we see the conversion of glucose 1-phosphate to glucose 6-phosphate. It also shows that the glucose 6-phosphate can either enter the glycolytic pathway through glycolysis, or it can be dephosphorylated, so you see the removal of the phosphate group here, and converted to glucose in the liver. This slide outlines the purpose of glycogenolysis in the muscle and in the liver, with only the liver being able to release the glucose cleaved from glycogenolysis into the bloodstream. So you see here that glycogenolysis in the liver allows for the release of the dephosphorylated glucose, so the free glucose, to enter the bloodstream. Whereas in the muscle, the free glucose stays within the muscle cell and is able to undergo the process of glycolysis. Glycogenolysis, and more specifically the activity of the phosphorylase enzyme, is highly regulated by either covalent or allosteric regulation. Interestingly, we also see that activation of the phosphorylase enzyme inhibits glycogen synthase, which is the key enzyme of glycogenesis. This diagram outlines the two regulatory processes regulating the phosphorylase enzyme. We see the process of the covalent regulation and the allosteric regulation. We will first review the covalent regulation. First, in this portion of the diagram, you will see the two forms of phosphorylase. We have first the inactive phosphorylase B form and the active phosphorylase A form. The difference between these two forms is that the phosphorylase B, which is inactive, is not bound to a phosphate group, and the phosphorylase A is bound to a phosphate group. So for the phosphorylase enzyme in the covalent regulation, the inactive form of phosphorylase is dephosphorylated, whereas the active form is phosphorylated. You can see that the phosphorylase B kinase enzyme catalyzes the reaction of adding a phosphate group to the phosphorylase B, which again is inactive, to form the phosphorylated active phosphorylase A enzyme. In turn, the phosphoprotein phosphatase enzyme removes the phosphate group, making the enzyme inactive again. The conversion of phosphorylase B to phosphorylase A is regulated by the activity again of phosphorylase B kinase. It will be important to note that there are a number of metabolic signals promoting the activity of phosphorylase B kinase. These metabolic signals include glucagon, epinephrine, cyclic AMP, and also calcium ions. It would make sense that these metabolic signals promote the activation of phosphorylase B kinase and eventually the phosphorylase enzyme, because these metabolic signals are elevated in a fasted state or when the demand for ATP synthesis is increased. For example, the hormone glucagon is elevated during fasting when blood glucose levels drop, and epinephrine, cyclic AMP, and calcium are increased during exercise. We will now review the allosteric regulation of phosphorylase. Here you will see there is only one form of phosphorylase, phosphorylase B. We see phosphorylase B inactive, and we see phosphorylase B active. In this type of regulation, phosphorylase does not form a covalent bond with a phosphate group or any other group. Instead, it temporarily binds an allosteric activator or an allosteric inhibitor to what's called an allosteric binding site on the enzyme. This temporary binding to the allosteric binding site changes the enzyme's orientation and thus its activity. AMP is the main allosteric activator of phosphorylase, converting phosphorylase B in the inactive form to phosphorylase B in the active form. And you also see that here, that phosphorylase B is regulated positively by AMP. We see that the allosteric inhibitors would be ATP and glucose 6-phosphate. This last slide summarizes the inhibitors and/or activators of phosphorylase enzyme at the liver and the muscle. We see here that the liver is activated by AMP through allosteric regulation and by glucagon through covalent regulation. The muscle is also activated by AMP and glucagon, but also cyclic AMP, epinephrine, and calcium ions through covalent regulation. It is allosterically inhibited by ATP and glucose 6-phosphate.